Welcome everybody. Thanks for the MLCSB COSI to host our proceedings contribution. As the title suggests, I'm going to present a method that we developed in order to do pseudotime analysis for single-cell time series data. This is going to be a method that will be pleasantly simple, I hope. All right, so as the background, we're talking about single-cell RNA sequencing-based studies. In the early days, typical experimental studies using single-cell RNA sequencing were based on a single experiment where a single cell suspension was analyzed by single-cell RNA sequencing in order to understand what kind of different cell types and cell states are present in the originating sample. And then maybe more ambitiously, to use this single-cell RNA sequencing experiment in order to derive trajectories of differentiation processes, or more generally dynamic processes that govern the variability in the single-cell RNA sequencing data. And then with the experiments becoming cheaper but still sufficiently expensive, studies started to perform multiple single-cell RNA sequencing experiments, possibly across multiple conditions, in order to then go ahead and define cell types and cell states using differential expression analyses, and then also carrying on to do pseudotime. And in this vein, another development was the upcoming of experimental studies that had a sequential experimental design. And most naturally, these types of studies are time series studies, where one tries to follow a biological process that expands over longer periods of time in a time series setup. And there, analogously, the goal or the types of questions that you have for this type of data is to identify genes that vary with time, and also to come up with a cell ordering in a similar fashion as these pseudotime trajectory inference approaches, to come up with a cell ordering that respects the time of the time series experiment. And one of the first data sets that has been generated that has this time series setup was a data set from the Quake lab. And in these early days, I think still done with a Fluidigm C1 chip, covering 411 acinar cells from the pancreas, from 8 different donors that had different ages, from 1 to 55 years. And in this study, one way to go ahead and perform coming up with some sort of pseudotime, identifying genes that vary with time, was to perform principal component analysis and then look at the first principal component as a proxy of this pseudotime. And what you can see on the right hand, or like on the left hand side, is this PCA analysis, the first 2 principal components of this data set, the data set projected onto these 2 dimensions. And on the right hand side, you see a distribution of cells coming from the different samples, from these different time points, from these different ages, with respect to the PC1 coordinate. And if the PC1 coordinate was a coordinate that well respected the time series labels, then what you would like to see is no overlap, or at least little overlap of cells that come from the same sample. And this is clearly not the case. And you might think that if you now take a real pseudotime ordering approach like Monocle, that is able to, or that at least for many single-cell RNA sequence data sets, is able to come up with a better cell ordering that respects the time of the underlying biological process, that you would get a better overlap or lack of overlap of cells from the different time labels. And what you can see on the right-hand side, that this is actually not the case at all. Actually, it's worse as the distribution of the data with respect to the first principal component. And the reason for this is that all these approaches like the simple PCA approach or these more sophisticated pseudotime approaches, are unsupervised approaches that are governed by the global variability in your data. And what we try to do here is to identify axes of variation that are confined to the variation with time. And so for this reason, we came up with a method, excuse me, that uses the time label information in order to come up with such a pseudotime that respects time. And for this reason is supervised. And so here you see an overview. Maybe I just get a pointer to guide you through this overview here. So you start off with a single-cell RNA sequencing data matrix where you have rows corresponding to cells and columns corresponding to genes. Then every single cell is assigned a time label. And the time label of the cell corresponds to the time label of the originating sample. And then by virtue of ordinal logistic regression model, we then go ahead and identify genes whose expression levels are associated with the evolution of time, and that also come up with a pseudotime ordering as a second result. And so how does this work in more detail? As the name suggests, the method is essentially a logistic regression model. And in fact, it's a logistic regression model, or it's a set of parallel logistic regression models that try to do classification of samples with respect to every single time point that has been considered in this study. And the input of this logistic regression model are the transcriptomic profiles of the individual cells, and the model defines a probability, a likelihood that is based on this logit transformation. And this is a logit transformation of a linear combination of the measured genes, modulo and offset, that is specific to the classification task instance. And as I said, this ordinal logistic regression model is a parallel set of logistic regressions defined for every single time point in this study. And every single of these logistic regression models has a shared parameter vector beta that puts or defines importance weights for individual genes. And this linear combination, beta times the transcriptomic profiles of the single cells, can then be used as a pseudotime that is associated with the time that you try to associate with. And by virtue of performing a sparse ordinal logistic regression using an L1 penalty, the parameter vector beta is enforced to be sparse and therefore be more interpretable. And now if you run psupertime, this approach we call psupertime, on this set of acinar cells, then with respect to the psupertime, to the supervised pseudotime that we define, the separation of the cells coming from the individual samples is a lot clearer than for the approaches before. And you might say, why is the separation not perfect? And the reason for this might well be that the processes that we look at are asynchronous, and therefore you might expect for many biological processes not to achieve this perfect separation of these distributions. And now you can go ahead and analyze which kind of genes are actually important to perform this ordinal logistic regression. So you investigate which of the genes get weights in the parameter vector. And here you see the 6 most important genes, the 6 genes that get the highest weight. And there you see a few usual suspects that make a lot of sense in the context of analyzing the maturation of acinar cells. So for instance, you see the gene encoding for alpha amylase popping up as being important. That is a known maturity marker of acinar cells. You see, for instance, clusterine as the second most important gene. This is also a gene that is known in the context of pancreas regeneration. And then ITM2A is a driver, is a gene that is known in the context of chronic pancreatitis and has been developed, reported and discussed in this context, but not so much in the context of the development of acinar cells, which might give rise to further follow-up studies to further investigate the role of this gene for acinar cells. In order to verify whether this ordinary logistic regression approach is actually helpful, we benchmarked this approach with a few other data sets, other single-cell RNA sequence data sets, studying human germline cells, embryonic beta cells, human ESCs, and the transition of mouse embryonic fibroblast cells to neurons. And what we evaluated is the Kendall's tau of the respective time series experiments, or like the time labels from these time series experiments with the pseudotime that you can come up with using psupertime or the baseline approaches. And as baseline approaches, we used PCA again, and then two very popular pseudotime ordering approaches, monocle and slingshot. We also compared to an approach that came out very recently that also uses time series information to perform trajectory inference, namely Tempura from the Bader lab. And what you can see is that psupertime across the board performs best, or at least very competitively, in particular in comparison to the unsupervised approaches. You see that for two of the benchmark data sets, psupertime performs also better than this time series dedicated model Tempura. And these are two data sets that, so let me just see, this is the embryonic beta cell data set, that's this data set here in the second row. And it's also this data set for the human germline cells. And maybe in particular for the human germline cell data set, you can already see what might be the reason why these supervised approaches perform better than unsupervised approaches. And these are situations where the global variability, in this case of different cell types, is much larger than the variability induced by the time series. And in the last few minutes that remain, I wanted to briefly touch upon a different application for this psupertime approach. And that are situations in which you do not actually have a time series experiment, you physically perform a time series experiment, but instead you might want to introduce manually user-defined sequential labels. So for instance, you perform a single-cell RNA sequencing experiment and you perform trajectory inference, pseudotime ordering, and then because you know your domain very well, you can come up with a sequence of cell groups, of clusters that corresponds to some sort of temporal measure. And so an example that we looked at in our paper was a data set, a single-cell RNA sequencing data set of colon cells that recapitulate the differentiation of stem cells in the respective epithelia to colonocytes and goblet cells. And by performing unsupervised clustering and also, I mean, looking up the underlying study, we could go ahead and define sequences of clusters that correspond to different branches of this differentiation process into these terminally differentiated cells. And for each of these branches, now you can perform psupertime analysis in order to figure out which genes are correlated with the evolution along these ordered clusters. And so here you see an overview of the most important genes that pop up from this analysis, also involving usual suspects. And then something that one can also do on top of identifying individual genes is to identify genes that have similar temporal profiles by just clustering the set of genes that come out of the psupertime analysis. And for these sets of genes, perform gene set enrichment analysis, gene ontology analysis, in order to identify what kind of processes are represented by these gene sets. And with this, I'm already at the end. I wanted to make you aware that there are these types of single-cell RNA sequencing studies that have this time series information. And I hope I could convince you that these types of studies benefit from a dedicated approach that takes into account the sequential information. The type of approach that we propose is a supervised approach. This approach specifically is based on a sparse ordinal logistic regression solution. It provides you with a cell ordering and pseudotime. It provides you with a sparse set of time-varying genes that you can then further analyze as you wish, as for instance, performing these kinds of gene set enrichment analyses. What we could show that, at least for the data sets we considered in this study, it was of great benefit to include this time series information in order to identify the ordering and the set of relevant genes. And other applications besides physical time series experiments are these studies where users define sequential sets of cell groups that can then be analyzed with psupertime. And we believe that these kinds of analyses will become more and more important, considering all these cell atlas approaches, cell atlas initiatives that come up with this larger and larger data sets. And last but not least, acknowledgements to the people that did the work on the ground. Will McNair, a PhD student that left my group a while ago, actually, whose baby this work is that I now have the pleasure to present. And then Revan Gupta, who helped a lot with the revisions. Thanks.
Thank you, Manfred, for this great talk. We have questions. Let's start with them. Thank you.
Okay, so thank you all for the great talk. So I have questions. I just wonder, so your data set is from the single-cell RNA data from donor at different age. I just wonder if your method can be applied to a patient cohort. For each patient, we also have a time series of single-cell RNA data. Do you have any suggestions?
Yes. So in principle, yes. Yes. In principle, yes. So all the data sets that we used in the benchmark that I glossed over maybe a little bit too fast are all data sets of the type that you describe. So these are more like these time series experiments where the duration or like the different difference between the different time labels is actually smaller, like days, months, minutes, or something like this. But yes, I mean, the short answer is, in principle, as long as your single-cell RNA sequencing data is sequential, you can just apply psupertime and get these types of results.
That was great. I'm not sure if this is actually. There we go.
Okay.
So, yeah. I think the idea makes a lot of sense, and I like it. And my one concern though is I see from the paper you did cross-validation to select your hyperparameter for lambda,
Yeah,
right,
yeah.
for the regularization weight.
Yes.
Yeah. I'm worried that you need another level of cross-validation for the parameters because you're doing supervised training on a data set and then using that same model on the same data. So have you done a control experiment where you permute the time labels and try to do this and make sure that it doesn't order them? Because it seems like you have to have control.
I mean, so first of all, I mean, the cross-validation that we do in order to well, not overfit, leaves out complete time points in order to avoid this. And if I'm not mistaken, Will did this permutation experiment that you just mentioned. And I think it went well. And it should be in the supplement. But don't name me on this. Maybe offline we can have a look at this. There's certainly a very important comment. Yeah. Thanks.
I have 2 questions. First, do we need to have any biological replicates in order to use this approach? I mean, like two single-cell data sets from the first time point, two from the second, and so on. And another related question, if I have, let's say, 4 time points from treated condition and 4 from untreated, is there a way to see the differences between this time series and another time series?
Yeah. So maybe regarding the first question, I think it's always helpful to have replicates because you obviously always suffer from batch effects across different samples. And so when we did this analysis here, we actually didn't do any batch correction. And I mean, we don't feel so bad about not having done batch correction here, because at that time, batch correction was not such a big issue, is because we have to have really bad luck to still like have false positive discoveries because of batch effects, because we regress over so many different time points. So we must really have bad luck to have a systematic batch effect that is associated with the time labels in order to suffer from that. And so, but still, I mean, having replicates would certainly be always helpful to also be shielded against this issue. And then the second question was related to comparing time series.
Is there a way to see the difference between two sets of time series experiments?
Yes, yeah. Well, I mean, you would have to compare the parameter vectors of the regression model, like the betas, and there, I guess you get some estimate about the confidence and the variability of the entries of this parameter vector beta for one time series and for the other, and then you can do some sort of hypothesis test that allows you to identify, well, differences of the loadings of that vector.